Have you heard about genetic mutation? Different eye color. Two-headed animals. Excess hair. Extra fingers. Mutations are changes in the genetic sequence, and they are a main cause of diversity among organisms. These changes occur at many different levels, and they can have widely different consequences. In biological systems, they are capable of reproduction. We must first focus on whether they're heritable. Specifically, some mutations affect only the individual that carries them, while others affect all the carrier organisms, offspring, and further descendants.
So, how do mutations occur?
The answer to this question is closely linked to the molecular details of how both DNA and the entire genome are organized. The smallest mutation are point mutations, in which only a single base pair is changed into another base pair. Yet another type of mutation is the non-synonymous mutation, in which an amino acid sequence is changed. Such mutations lead to either the production of a different protein or the premature termination of a protein.
Over a lifetime, our DNA can undergo changes or mutations in the sequence of bases A, C, G, and T.
This results in changes in the proteins that are made. This can be bad or a good thing.
Mutations can occur during DNA replication if errors are made and not corrected in time.
Mutations can also occur as a result of exposure to environmental factors, such as smoking, sunlight, and radiation.
Often cells can recognize any potential mutation, causing damage, and repair it before it becomes a fixed mutation. Here we have a video about mutations in DNA and how can they make mistakes.
DNA rarely makes mistakes, but it can happen. A point mutation changes a single base. This can change the whole amino acid, or depending on its position, could go unnoticed. Other mutations can be more damaging. A deletion removes a base, but also causes a frame shift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the deletion, the amino acid sequence will change because the bases shift by one. The resulting protein will likely be non-functional. An insertion adds a base, but also causes a frame shift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the insertion, the amino acid sequence will change because the bases shift by one. Again, the resulting protein will likely be non-functional. Lastly, an inversion will invert a segment of DNA end to end. Watch this section closely. Each base has been moved around. Again, these mutations have the potential to be harmful, but not all mutations have an effect on protein. Remember, during translation, a start codon must first be read to begin coding the protein. If the mutation happens before or after the protein coding sequence, there will be no effect on the protein. Mutations must occur in the coding section of DNA to affect protein synthesis. Otherwise, nothing happens.
Is a permanent alteration in the DNA sequence that makes up a gene, such that the sequence differs from what is found in most people. Mutations range in size. They can affect anywhere from a single DNA building block to a large segment of a chromosome that include multiple genes. We all start our lives with some mutations. These mutations inherited from your parents are called germline mutations. However, you can also acquire mutations during the lifetime. Some mutations happen during cell division, when DNA gets duplicated. Still, other mutations are caused when DNA gets damaged by environmental factors, including UV radiation, chemical, and viruses. We all have mutations. All of us all humans are mutants. And as we know, some mutations are bad for us, but others are good. And these mutations help you and us survive in this environment and in this world.
And this was all. This was our video about gene mutation. Thank you for watching. We hope you like it.